Great. And we're going to share our screen. Okay, so let's jump in right off where we left off. So on Tuesday, we were talking about how to make ATP, so a pretty important thing for the cell. And we were talking about how whenever we talk about ATP, we refer to it as phosphorylation. And that's because what we're doing is we're adding a phosphate group to ADP, so we're phosphorylating ADP to create ATP. So that's why we have, when we say we have 3 different ways to make ATP, they're just like 3 methods of phosphorylation. And when it comes to making or phosphorylating ADP into ATP, we have really, again, like I said, 3 methods, and the difference between all 3 of these is really just where the energy is coming from to form this high energy bond right here. So where are we getting that energy? So we talked about the first method of phosphorylation, which is substrate level phosphorylation, which is when we have some compound with a bond in it. And we break that bond, and as we know, electrons are associated with bonds. We say they're stored in bonds. And so when we break this bond, electrons are released, and that gives us enough kind of energy and a good source of electrons that we can store those electrons in that 3rd phosphate bond. So we can convert ADP into ATP. So it's important to note, right? So a lot of students get kind of hung up on the difference between substrate level phosphorylation and oxidative phosphorylation. And what I will tell you, the biggest difference here, right? So always, always, always look for an electron transport chain. With substrate level phosphorylation, with what we're looking for on this slide, we do not see an electron transport chain. ATP is made right here. It's called substrate level phosphorylation because it's made at the level of the substrate. There's no electron transport chain. It's one step. So that's substrate level phosphorylation. Energy is released from breaking a bond. A phosphate is released from the site of a reaction. But whatever it is, it's enough to make ATP right then and there. All right. And then we have oxidative phosphorylation. So I'm just going to go ahead and jump to our kind of summary slide for this because we did talk about this on Tuesday. And I actually like this slide the best for explaining it. So we know that oxidative phosphorylation requires 2 steps, whereas substrate level phosphorylation is going to form ATP right here, right where the electron is released. With oxidative phosphorylation, what happens is you have oxidation of this. So you still break a bond and release an electron. So that is oxidation. You have a paired reduction reaction. So remember, reduction is when a molecule is reduced. And so what happens is the molecule that is reduced is NAD+. And when it gains an electron, it also grabs up a hydrogen. And so it is reduced from NAD+ to NADH. So we don't write down that it's carrying the electron. In chemical formulas, we don't ever, it's not an element. It's just a subatomic particle. So that's why we see NAD. We do note that it grabs up a hydrogen because that's a molecule. And so NAD is reduced. When we say, again, it's reduced, that means its charge is reduced because it gained an electron. And we go from NAD+ to NADH. So what that means is we had this molecule with a bond. We broke that bond, released electrons, and now NADH is carrying those electrons. It's called an electron carrier. And it carries those electrons to an electron transport chain. So an electron transport chain requires a membrane. So we see a membrane right here. And what happens is once it gets to a membrane, NADH is oxidized, meaning it releases an electron, releases its hydrogen. And what it does is it uses the energy from that electron to pump hydrogens to one side of a membrane. So we left off looking at pictures of the Hoover Dam. And the reason I always bring the Hoover Dam into this conversation is because you can think of, so what happened here is we used an electron transport chain. So it's a chain where electrons are helping in the transport of hydrogens to one side of a membrane. So it's a transporting hydrogens. But once you get all those hydrogens on one side of a membrane, that's what we call a proton motive force. So remember, we call hydrogen ions protons because that's basically all they are. They're just like a proton. And so we have a lot of protons or hydrogens on one side of a membrane. So remember, when we have more molecules on one side of a membrane compared to another side of a membrane, that's a concentration gradient. And concentration gradients, because everything wants to be in equilibrium across a membrane, if you have a concentration gradient, that is a lot of potential energy. Again, think of all the water stored in Hoover Dam. That's a lot of potential energy. So what happens here is the electron transport chain generates a proton motive force. It pumps all the hydrogens to one side of a membrane, so you have a concentration gradient of hydrogens. Those hydrogens really want to get to the other side of that membrane. And the molecule that allows them to do that is ATP synthase, which you see here in purple. So basically what happens is every hydrogen that's able to fly through ATP synthase gives enough energy to convert or to phosphorylate one molecule of ADP into ATP. So our kind of equivalent calculation here is every one hydrogen that passes through ATP synthase will give us one ATP. So pretty amazing. So again, that's oxidative phosphorylation. It breaks a bond in a molecule. We release an electron. That electron is carried to an electron transport chain where it pumps, generates a proton motive force, and then that's what allows us to get ATP. All right. So again, 2 steps as opposed to 1. And then we have photophosphorylation. Photophosphorylation is very, very similar to oxidative phosphorylation. You can see in this picture, I apologize, it's kind of hard to see, but you have a series of molecules called the photosystems, which hopefully we'll talk about later. But what happens is you have an initial molecule in the case of photosynthesis, which uses photophosphorylation. Photo or light, photons from light are actually where we get enough energy to break our initial bond. So it's not an oxidation reaction. Well, it is an oxidation reaction. But in the case of oxidative phosphorylation, this is typically mediated by an enzyme right here. So typically we have an enzyme that uses or does this process. Whereas in photophosphorylation, what allows us to break that first bond is actually photons from light. So we absorb light, it energizes these, ends up splitting water, and electrons are released from the bond that is in water. So not only are electrons released, but hydrogen is also released. So you can see here that when water is broken, those 2 little teensy tiny molecules are electrons, and they're going to enter the photosystems, which we'll talk about later. But important thing here is that we have some hydrogen released. And you can see that chloroplasts are compartmentalized, very similar to mitochondria. And here we have the thylakoid space, which is sort of like on the interior of these membrane folds. And then we have the stroma, which is the exterior of these membrane folds, still inside the chloroplast. But you can see that because we've split water and released hydrogen, we've generated a concentration gradient. We have a ton of hydrogens outside of, or in the thylakoid space, sorry, and then fewer hydrogens in the stroma outside of that thylakoid space. You can see that we also have an electron transport chain here, pumping any exterior hydrogens into this space. So essentially what happens with photophosphorylation is same thing. We break a molecule, get some electrons, and those electrons are used in an electron transport chain to generate a proton motive force, which again, a proton motive force is just a concentration gradient of protons, hydrogen ions across a membrane. And we see that concentration gradient, we see all those hydrogens in the thylakoid space, fewer in the stroma. So that gradient, again, it's like the Hoover Dam inside the thylakoid disc. And so what starts to happen is those hydrogens start to try to get to equilibrium. They fly through ATP synthase, and we get ATP. Excuse me. These allergies can be the end of me, I swear. Okay, so the big difference here is that with oxidative phosphorylation, some sort of enzyme breaks this bond and releases an electron. Whereas with photophosphorylation, that bond is broken via light from the sun. You still have an electron transport chain. So notice that both oxidative phosphorylation and photophosphorylation utilize an electron transport chain. So that is very important. Both oxidative phosphorylation, oh my goodness, my nose. Ugh. Both oxidative phosphorylation and photophosphorylation utilize an electron transport chain. Substrate level phosphorylation does not. All right. So this is just a slide to summarize these three pathways of making ATP. And again, the big difference, there are some differences mechanistically, but the big difference is where the energy is coming from. So with substrate level phosphorylation, an energy from an exergonic reaction is enough. So we release electrons from a bond, and those electrons immediately go into the phosphorylation of ADP. We also have oxidative phosphorylation, and this is where we have an oxidative reaction. This is mediated by an enzyme. But instead of using those electrons immediately to generate ATP, we carry them to an electron transport chain and generate a proton motive force, and that is what is used to make ATP. So why would we do that when we can do substrate level phosphorylation, which is way easier? As you will see, oxidative phosphorylation has the capability of generating like 3 to 4 times as the amount of ATP as substrate level phosphorylation. It is vastly more efficient in energy generating. And then photophosphorylation is we're still breaking a bond, we're still releasing electrons, we're still doing an electron transport chain to generate a proton motive force. It's just that instead of using an enzyme to break that bond, we use light from the sun, which is why we say photophosphorylation. So oxidative phosphorylation and photophosphorylation are extremely similar. The difference is just what's breaking that initial bond. Okay, so, whoops. Forgot to activate the chat box. Hold on just a second. Okay, I have the chat box on, so if you have any questions at any time, feel free to ask. All right, so now that we understand our 3 different types of making ATP, or ways of making ATP, we're gonna go into our major energy generating pathways. So we're gonna start with cellular respiration. Which we're going to spend probably the majority of our time on, if not all of our time on today. Okay, and that's because all organisms do it. There are very few organisms, well, I will say nearly all eukaryotic organisms do this. I don't want to say all organisms do that. It's not true. As a microbiologist, I cannot say that. But it is one of the most ubiquitous pathways utilized by organisms, cellular respiration. All right, so we're going to talk again, like I said, about 2 major energy generating pathways. These energy, I guess, again, because of the law of thermodynamics. We really shouldn't say that these are generating energy, but these are our major energy transforming pathways. Let me just update that because we want to be accurate. There we go. So these are linked, as we will talk about in the ecosystem. We have organisms that perform photosynthesis and organisms that perform cellular respiration. In some cases, the same organism can perform both. So plants have both of these organelles. They can do both of these. So what happens is we have photosynthetic organisms, which are capable of transforming kinetic energy into potential energy. So they take kinetic energy in the form of the sun, and they transform it into potential energy in the form of organic molecules. And then what the mitochondria does is it transforms that potential energy stored in organic molecules into kinetic and usable energy in the form of ATP. So you can see that these linked are by their reactants and products. So cellular respiration, which is kind of demonstrated by the mitochondria, because that's where some of the steps of it occur, organic molecules and oxygen go into this reaction, generate ATP, and the byproducts of that reaction are carbon dioxide and water. But those are actually the reactants for photosynthesis. So for photosynthesis, you use carbon dioxide and water and energy from the sun, and what comes out of that reaction is organic molecules and oxygen. So these are our two major energy transforming pathways in cells, but they're also intimately linked. So we talk about these as some of our major metabolic pathways. Again, not the only metabolic pathway, but some very, very, very kind of like central pathways. All right. So I want to say one thing here, and I have to say it as a microbiologist, that we typically associate photosynthesis and cellular respiration with these membrane-bound organelles, chloroplasts in the case of photosynthesis, and a mitochondria in the case of cellular respiration. But as you know, we already talked about the endosymbiotic theory about how these organelles likely originated from the phagocytosis of these organelles by a larger organism, both originally prokaryotic organisms, meaning they didn't have membrane-bound organelles until this this phagocytic event happened, the endosymbiotic theory. So what I want to the reason I mention that is because I really want to hesitate for you to like think that if an organism does not have a chloroplast, or if it doesn't have a mitochondria, that they can't do cellular respiration or photosynthesis, because that is not true at all. The vast majority of the organisms on this earth, bacteria, that do photosynthesis and cellular respiration don't have these organelles, right? These are membrane-bound organelles, meaning they're only present in eukaryotes, which means prokaryotes, if those processes were dependent on these organelles, prokaryotes wouldn't be able to do cellular respiration or photosynthesis. And that's just, so what organisms do that don't have chloroplasts or mitochondria, again, the only reason these organelles are like great is because they're made of membrane. And prokaryotes don't have these membrane-bound organelles, but they have a plasma membrane. So that's actually what prokaryotes primarily use. Is if prokaryotes don't have a mitochondria, don't have a chloroplast, which, you know, like by definition they don't, then what they actually do is they just find modifications so that they can use their plasma membrane as a source of membrane for the electron transport chain. So I just think that that's really important to mention because, again, this is showing these organelles being kind of central to this process, but really, as I have hopefully emphasized since the beginning of this class, it's not these organelles that are central to these processes. These organelles are special because they're made of folds and folds and folds of membrane. Membrane is central to this process because it provides a location for the electron transport chain. So that's why I always emphasize that. I had a question. Great. The question is, do organisms that derive energy from things like volcanic vents use a similar energy production method, or do they have similar organelles? That is such a good question. And the answer to that question is the majority of organisms that utilize energy from like volcanic vents or like our extremophiles, those are going to be prokaryotes. So they're definitely not going to have a chloroplast or a mitochondria, but they will have a plasma membrane, and they will typically have a cell wall. And both of those things allow them to still be able to generate an electron transport chain and a proton motive force. So yeah, even those like crazy organisms living in volcanoes or deep sea thermal vents or, you know, Yellowstone or even like Antarctica and the glaciers, these organisms still have a membrane. So they can still do these processes if they need to. What I will say is that a lot of times those organisms actually use a different process. They use fermentation, which doesn't necessarily require that membrane. But that's not to say that they can't do it. Most of these prokaryotes have the genes that would allow them to do cellular respiration. But for prokaryotic organisms, because they live such a short amount of time, typically fermentation, which we'll talk about a little later, is sufficient. So they don't need to like make all the stuff to do cellular respiration. But you have a lot of the organisms that kind of cause problems. And if they're bacterial, or a lot of the environmental bacteria that don't cause problems that are really good, those ones tend to like to do cellular respiration if they can. Although bacteria tend to like fermentation quite a bit. All right, good, excellent question. All right, so let's get into it. So I do want to talk about some commonalities between our major metabolic pathways, cellular respiration and photosynthesis. So I talked already about when we were talking about oxidative phosphorylation, I talked a little bit about the fact that we use an electron carrier, and it's called NAD+. So that stands for, really, no one calls it this ever, but if you want to know what it's called, it's nicotinamide adenine dinucleotide, but that should tell you that it's made of some pretty good recycled material from a cell, which is adenine dinucleotide. Adenine is also present, obviously, in DNA, and nucleotides are present in DNA. And so this tells you that it's using those nucleotides that are kind of like freely available in a cell for whenever you need to do transcription or replication, they're there. So this molecule is NAD+, and anytime that it is reduced or gains an electron, it becomes NADH. So if you see NAD convert to NADH, that just means that it grabbed an electron, and it's probably carrying it to an electron transport chain. And we can actually measure, you can measure NADH to say how much, you know, glycolysis has happened, how much of the Krebs cycle has happened. You can measure NADH as kind of a product of those metabolic pathways because it's generated so freely through those. And kind of compare NAD to NADH. That's how we do a lot of metabolic experiments. And then we have flavin adenine dinucleotide, just kind of a different, instead of having an amide group, it has like a flavin group. And then, so that's called FAD, and that actually carries 2 electrons and it becomes FADH2. So, so this says it's carrying an electron. But these, essentially, we don't really even need to know the chemistry behind this. You'll get that eventually in biochemistry, but what I want you to know is that these molecules hang out around reactions. So when a bond is broken and electrons are released from that bond, these are the molecules that are reduced and carry that electron to the electron transport chain. So that's for cellular respiration. In photosynthesis, we use a different electron carrier. We use NADPH. So that's pretty much NAD, but with a phosphate group. And then when it is reduced, I think I said NADPH. Well, it's NADP. And when it's reduced, it becomes NADPH. And again, that hydrogen just tells you that it's carrying an electron. We also grabbed a hydrogen to sort of neutralize it. So those are our major energy, our electron carriers. For cellular respiration, we use NAD and FAD, and we, as our molecules that we reduce. In photosynthesis, we use NADP. And then importantly, in both cellular respiration and photosynthesis, both of these are going to use these electron carriers to carry electrons to an electron transport chain and generate a proton motive force. So both of them use what's called chemiosmosis, which again is just using an electron transport chain to generate a proton motive force. And both of them will use that electron transport chain to fuel ATP synthase and basically charge the molecular battery that is ADP into ATP, which is usable cellular energy. All right, so let's get into it. Okay, so this is a very important pathway. I really want to make sure you understand this. So we're going to go through it today. I also would recommend watching the pre-recorded lecture I made. Do those Learn Smarts. And like I mentioned at the end of lecture, we will also have kind of just like another chance to get together. I'm going to have sort of a special study session for all of you. Okay, so with cellular respiration, that overall reaction is that we take an organic molecule. So we typically think of this as being glucose, although it doesn't have to be. But we typically think of this as being glucose, so it's chemical reaction C6H12O6. So importantly, it's an organic organic molecule because it's got all three of those, carbon, hydrogen, and oxygen, linked by chemical bonds. We also use some oxygen. So I want you to look at the reactants going into this. We have 6 carbons. And then we're going to have 6 oxygens utilized. And you'll notice that our first product listed is 6 carbon dioxides, which is carbons and oxygens. So you can see what we're going to do via cellular respiration is we're going to break down glucose. But as we start breaking off those carbons, we're actually going to combine them with oxygens. And that's where we get the release of carbon dioxide. We're going to release those water and the hydrogen and oxygens as water later on. So that's where that part of it goes. And then we also generate a ton of ATP. So we generate this ATP via 2 primary methods of ATP generation. Cellular respiration uses both substrate level phosphorylation and oxidative phosphorylation. We will look at our energy totals later. The vast majority of energy in cellular respiration is generated via oxidative phosphorylation, but there are 2 events where it uses substrate level phosphorylation as well. We'll look at that. One of those is right off the bat, so we will look at it soon. The overall goal of cellular respiration is to take a carbohydrate, a sugar, carbon ring, break bonds in that carbohydrate, release electrons from those bonds, grab those electrons with electron carriers, and carry them to an electron transport chain where they will generate a proton motive force, and that proton motive force will be utilized to create ATP. So basically the overall goal is just to like send glucose through a giant oxidative phosphorylation reaction. which does very effectively. But because it's glucose, right, when we looked at oxidative phosphorylation, where is it? When we looked at oxidative phosphorylation, we were zooming in on just like one bond being broken, right? We're breaking this bond and releasing an electron, and that goes to an electron transport chain. What cellular respiration is trying to do is do this with a giant sugar. So it requires a lot more steps. This is just looking at a summary of these steps. I will revisit this panel picture later because I love it. I think it is an excellent summary. But you can see here that overall cellular respiration can be divided into 4 steps. Our first step is highlighted in yellow, which is glycolysis, where we take our 6-carbon sugar and basically just break it in half. Okay, so glycolysis, lysis means split, glyc is referring to a sugar, so glycolysis is splitting of a sugar. So we take our 6-carbon sugar and break it in half, and if you're breaking a 6-carbon molecule in half, you're creating 2 3-carbon molecules, which is where you get that pyruvate. You'll notice that coming off to the left, we do get some ATP generated right away. If you get ATP generated right away without an electron transport chain happening, such as what you're seeing right there, substrate level phosphorylation. So during glycolysis, you do get one event of substrate level phosphorylation. So you break glucose in half, go from 6 to 3-carbon, release a little ATP already, and then, but also you're releasing electrons from those bonds, which is why we see NADH released, which will go down to an electron transport chain. So it's important though to follow along to what's happening to our organic molecule. So we split 6-carbon glucose into 2 3-carbon pyruvates, and now we need to continue to break those down. So those pyruvates, those 3-carbon molecules, enter the second stage of cellular respiration, which is called the intermediate stage, which is not really shown on the slide very well. But the intermediate stage, you also get some NADH release. I don't know why it's not shown here. You get some NADH released. And then after the intermediate stage, you do enter the Krebs cycle. You'll notice, what do we see right here? A little ATP. That's because the Krebs cycle also has a little bit of substrate level phosphorylation. But primarily you can see we get a lot of NADH, a lot of FADH2, which should tell you that oxidation is happening, reduction is happening, and all of those electrons released during glycolysis, during the intermediate stage, during the Krebs cycle, are going to go to the electron transport chain, where we're going to generate some ATP. And then I also want you to pay attention to the fact that after the electron transport chain goes through, we're going to have some like extra electrons hanging out. And so oxygen, also some extra hydrogens hanging out. And what we do with the electrons and hydrogens, basically after we're done with them from the electron transport chain, is we bind them up to oxygen and we make water. All right, water is what I said. And you'll see that the overall process of I should have mentioned that this is called aerobic because it's referring to the fact that oxygen is used as that final electron acceptor. Basically, oxygen comes into the electron transport chain to clean everything up. So that's aerobic cellular respiration, and you'll notice that it overall produces 38 ATP. That is a lot of ATP. That's awesome. All right. I did also want to mention where this takes place in eukaryotic organisms. Glycolysis takes place in the cytoplasm, but after glycolysis is complete, pyruvate, that 3-carbon molecule, actually enters the mitochondria, and the remainder of cellular respiration actually takes place in the mitochondria. So you have glycolysis occurring in the cytoplasm, meaning you're getting a little bit of ATP generation in the cytoplasm right away, but the majority of the electrons are carried by NADH and FADH2 into the mitochondria. We have some more electrons released here and during the Krebs cycle, and then these enter an electron transport chain, which also takes place in the mitochondria. So what you can see here, again, is we do get a little bit of ATP released via substrate level phosphorylation, right? Little ATP via glycolysis, little ATP via the Krebs cycle, but we also see up here that we are oxidizing compounds and releasing them electrons, and those electrons are being carried to the electron transport chain. And you can see overall that this like star or like bang of ATP is way larger from oxidative phosphorylation. So during cellular respiration, we do get a little bit of ATP generated during glycolysis via substrate level phosphorylation, a little bit during the Krebs cycle via substrate level phosphorylation, but the vast majority, and I mean like 34 out of the 38 ATP generated during cellular respiration, if I remember correctly, 32 to 34 of the ATP generated via cellular respiration is via oxidative phosphorylation. So it's a very powerful process. I had a question. The question is it possible for something other than oxygen to serve as a final electron acceptor? And the answer is absolutely yes. Good question. We'll get there. So let's talk about aerobic cellular respiration first, just kind of so we can see how oxygen serves as a final electron acceptor. But for organisms that are not in the presence of a lot of oxygen, absolutely, you don't have to use oxygen as a final electron acceptor. In fact, not even humans always use oxygen as a final electron acceptor. We use some other stuff as well. And another question, are neurons specifically made to conduct glycolysis since the brain consumes a large portion of the body's glucose? Let me see if I can answer that question. So I would say that in the human body, the vast majority, I want to say like pretty much all of your cells in your body, pretty much all of your cells are suited to absorb glucose and immediately send it through glycolysis if it needs to create energy. And what I mean by that is, as we'll see later, and we kind of talked about before, we don't always need a ton of energy. So instead of sending glucose through cellular respiration, we can store it in the liver. But also, we may not need a ton of energy. We may need more like macromolecules. And as we will see a little bit later, cellular respiration is not only vital for the generation of ATP, but it also plays a really important role in macromolecule generation or biosynthesis. So in that case, I would say that neurons for sure, because they do use a lot of energy, but every single cell in an organism is going to be able to perform aerobic cellular respiration and anaerobic cellular respiration and fermentation. They can all do it. Not that they will always be doing it at any given time. Actually, interestingly, I don't mean to brag, but when my husband was a scientist, maybe he still is a scientist, I don't know. He's in med school now. But my husband's research was on tumor cells. And specifically, he was looking at like leukemia. And what he found is that tumor cells, so cells that have kind of genetically made the switch to being immortal, those cells switch from doing cellular respiration to doing kind of like glycolysis only. So which is basically fermentation. So they do a lot of glycolysis, but they don't go through cellular respiration. Interestingly, that's also characteristic of some virus-infected cells. Not that there's necessarily a link between like leukemia and virus. It's just what that tells you is that the vast majority of your cells under homeostatic circumstances will, if they need energy, go through cellular respiration. They'll go through all 4 of these steps. But if homeostasis is sort of like disrupted, let's say like in a tumor cell or in a virus-infected cell, or maybe like a muscle cell that you've been exercising a ton and it's sort of like, I'm out of energy, that also switched to glycolysis. So that's a really good question. I would say that all of your cells can do cellular respiration, meaning that glucose can immediately enter glycolysis. But whether it proceeds through all 4 steps is situational. That's a good question. All right. Science is cool, man. All right. So this is just looking again. Please, please, please promise me you will remember that in cellular respiration, we do both substrate level phosphorylation and oxidative phosphorylation, and sometimes these can occur simultaneously, as we will see. So those are not completely divided reactions. You can see glycolysis, we have a little ATP generated via substrate level phosphorylation, but during glycolysis, we are also oxidizing glucose and releasing electrons, which will move to an electron transport chain and generate ATP. So those 2 processes, oxidative phosphorylation and substrate level phosphorylation, they don't happen necessarily independently, as we will see. A lot of times they happen simultaneously. Okay, so let's jump into the process. We've been summarizing it and we will continue to summarize it, but let's look at each of these steps independently. So the first step of cellular respiration is glycolysis. So glycolysis is an extremely important step, not only because it's the first step in cellular respiration, but also the process of fermentation, which hopefully we'll have time to talk about. Fermentation is literally only glycolysis. So anytime you have a cell that goes through fermentation, or you talk about bacteria or yeast fermenting food, or like I said, like a tumor cell or a virus-infected cell, when we say that a cell is going through fermentation, that means that it's going through glycolysis, but it's not going through the rest of cellular respiration. So why can it do this? And the answer is because of substrate level phosphorylation. Because a couple of things happen here. So let's look at the process of glycolysis. So again, glycolysis literally means splitting sugar. So you see the 6-carbon glucose right here. 1, 2, 3, 4, 5, 6. The first thing that happens during glycolysis is we use an enzyme to split glucose into 2. So that's anytime you see like my lightning bolts, that means a bond is being broken. So we see that in glucose, we actually have, because we're going from a 6-carbon molecule to 2 3-carbon molecules, we actually have 2 bonds broken. So a couple of things happen here. You'll notice that I actually have here, it says +2 ATP. That's because glycolysis is actually the only time during cellular respiration where we actually have to use some ATP to get this started. So glycolysis is, splitting sugar in half requires a lot of energy. This requires a lot of energy. So we're going to throw 2 ATP at it to kind of like give it that energy. So glucose, what we do, again, we take a 6-carbon glucose, we break 2 bonds in it. That requires 2 ATP. But you'll see the result of this is that we effectively split 2 glucose. We have 2 3-carbon compounds, and these are called pyruvates. A couple of other things happened. As you know, every time we break bonds, we release electrons. So we know electrons came out of each of these bonds. So you can kind of equate this to we break this bond, release one electron, and that one electron will be bound up by NAD+, meaning we generate one NADH for every bond. So we break this bond, electron is released, NAD grabs it, and we get one NADH. That happens to this bond as well, so we get an electron released, NAD binds it, and we get another NADH. So that means that during glycolysis, because we break two bonds, we get two NADH. Think of that, every time we break a bond, we get an NADH. Another thing that happens though is that the splitting of glucose, these bonds, because they're bound in a sugar ring, which is very, very stable, sugar rings are very stable. Because this is in a ring, there's a lot of energy stored in these bonds. There's also some phosphate, which we don't really talk about. But you will learn later. But another thing that happens when we break these bonds is enough energy is released that, look, we get some ATP. So not only did we release electrons from those bonds and get NADH, we also got ATP. So again, here's a very important thing. If you have NADH, that means glycolysis will contribute to oxidative phosphorylation. We're gonna eventually be able to use this to generate ATP. But glycolysis also directly, right here, generated some ATP. If we got ATP right here, right now, without an electron transport chain yet, that means we got it via substrate level phosphorylation. So another thing to note with glycolysis, the way this is sort of calculated, so you'll see on my top right, I have an energy tally. This is the overall energy that we've generated, and we'll kind of have this as we go through the process. I say that we've generated 2 ATP. And I say we've generated 2 ATP, even though technically we get 2 ATP per bond, which is why our result is 4 ATP. Technically, the way a lot of textbooks calculate it is they look at the energy tally as sort of like the overall gain of ATP. And since we had to use 2 ATP, technically if we take away the 2 that we used, that means our overall gain is only 2. So the way that they kind of refer to that is that they would say, yeah, there's like 4 ATP generated, but there's a net gain, so like an overall increase only of 2, because you had to put in those 2 at the beginning. So that's why you see in our energy tally, we gained 2 ATP, because even though we gained 4, we had to use 2. So we kind of subtract that initial 2 we utilized. So during glycolysis, we split glucose in half into 2 3-carbon molecules. We release some electrons from each bond, 1 electron from each bond, which is why we get 2 NADH. And then we also released enough energy to generate some ATP. We generated 4, but because we had to use 2, that gives us a net gain of 2 ATP. My husband is just waking up. Oh my God, lazy. Just kidding. Are you getting coffee?
Yeah.
Okay, so I had a couple of questions. Great. Okay, so one question was, if glycolysis takes place in fermentation, that what happens to the buildup of pyruvate? That is an excellent question, and I hope that we have enough time to answer it, but just in case. If this is only fermentation, what would happen is the problem here is not necessarily pyruvate, because we can use that for other things, but an issue is that in order to accept electrons, we need NAD+, but you'll see that this process requires NAD+ to be oxidized and generate NADH. So one of the problems with fermentation is you keep going through glycolysis, you're using all the NAD+, you're building up a ton of NADH, but there's no electron transport chain to use that NADH. So how do we get back the NAD+? And what we do for fermentation, or sorry, what we do for fermentation is we actually, weirdly enough, we oxidize NADH, and the electron is donated to pyruvate. So this becomes pyruvic acid. When it gains both the electron and the hydrogen, it becomes an acid. So this is why, and then some other reactions can take place. The body kind of handles that acid in a lot of different ways. But if you think about like working out, when you work out, something that starts to happen is as you burn through the glucose in your muscle, first off, you're doing cellular respiration to generate a ton of ATP. But eventually you actually run out of oxygen. So another thing you'll notice about glycolysis is that there was no oxygen involved in this process. So this can happen without oxygen. So you do cellular respiration when you're lifting weights, you're using cellular respiration for energy until your muscles run out of oxygen, which happens pretty fast. And then you switch to fermentation, which is just glycolysis because it doesn't require oxygen. But the thing here that starts to happen is as you do that, because you don't have an electron transport chain, that NADH is oxidized and it reduces pyruvate into pyruvic acid. So this is when you're lifting weights, when it starts to like burn, that's because you're having some accumulation of acid in your muscles. And then obviously when you think about what happens is like as acid builds up, then you can eventually get denaturation. So obviously your muscles are not going to denature, but what starts to happen is as that acid starts to build up, the homeostasis in your muscles starts to kind of like be disrupted. So this process can't take place as easily. So that's when you get to the point where you like literally can't lift weights anymore, to the point of just full exhaustion. But if you lift weights, you'll notice that, you know, like you can recover from that relatively soon, and that's because you breathe and you replenish your muscles with oxygen. So it's an excellent question. Another question, is that why some fermented foods taste like pepperoni and salami, have high levels of lactic acid? Exactly. Any fermented food is going to have sort of like a tart, kind of sour, spicy flavor, kind of texture. Kind of think about like kombucha kind of being sour, or kimchi sort of having that sour flavor. Any fermented food is going to have a lot of acid in it, and it's because that's a byproduct of fermentation. Good job. Yeah, it's such cool stuff, you guys. I love this stuff. Let's extend class for a couple of weeks. Just kidding. I don't have that power. Otherwise, I would say let's do it. Okay, so which is maybe why you don't want me in charge. Okay, so let's talk. We talked about glycolysis. What we did with glycolysis, we took 6-carbon sugar, we split it in half. So we're going to go through cellular respiration. So if we go through glycolysis and split it in half, we get 2 pyruvates. We generated a net gain of 2 ATP, and we also released electrons from each of those bonds broken, bound them up with NAD+, and created NADH. So you can see at the bottom of the slide, what went into this reaction was we did expend some energy, but a 6-carbon glucose. And then the products of this reaction are 2 3-carbon molecules of pyruvate, 4 ATP, although you can see in our energy tally, we say that's a net of 2, because we only really gained 2, because we used 2. And then also some NADH, which we can use in an electron transport chain. All right. So now our question is, what happens to this pyruvate through cellular respiration? And the answer is, it enters the intermediate stage. So the intermediate stage is called like so many different things. Sometimes it's called acetyl-CoA production. sometimes it's called pyruvate oxidation. I mean, I wish that, you know, like science would get a consensus on what to call this, but currently everybody kind of calls it something different. I like calling it the intermediate stage, probably because that's just how I was taught. But pyruvate oxidation technically makes a lot of sense. So the biggest thing here is just, I would recommend just knowing what happens. So after glycolysis, you have 2 3-carbon pyruvate molecules. And the only thing that happens to the, 2 major things happen to the pyruvate. So you'll notice that in this picture, I diagrammed just what happens to one of those pyruvates. But remember, through glycolysis, we have, oh my gosh, my fingers. Oh, we have 2 pyruvates. So this is happening to both of those pyruvates. So we have 1 pyruvate here. We are always going to be breaking bonds. So you can see that the bond here is broken. So we know that if a bond is broken, we release electrons, and that's why we get some NADH. Okay, so we got some NADH because electrons were released. Something also that is new here compared to glycolysis, so we broke this bond, and you'll notice that what is breaking off is a 2-carbon molecule, which is an organic molecule, so that will go forward. But what also is breaking off is a single carbon. So our body is really good at recycling. And one thing that it definitely wants to recycle is carbon, right? Because carbon's the building block for all organic compounds. Also, carbon is a really good reactant for photosynthesis, which builds organic compounds. So what happens is we break this bond, and we are going to use our resultant 2-carbon still organic compound that will kind of move on to the next step of cellular respiration. But the body does, or a cell does, want to be sure that it can recycle this kind of like free carbon. So it does that. Notice that this is the first step in cellular respiration. At no point in glycolysis did we need oxygen. But during the intermediate stage, we need oxygen because what we're doing is we're releasing 1 carbon, and we bind up that carbon with oxygen, and that makes CO2. So anytime you see a product of carbon dioxide through cellular respiration, that tells you that that step requires oxygen. It also tells you that a carbon is being released. So again, glycolysis, we don't need any oxygen to do glycolysis. The big reason we don't need any oxygen to do glycolysis is because we're not releasing any carbons. We just broke an organic compound into like 2 organic compounds. There's no carbons released. But you get into the intermediate stage, and because we break this bond, we get the release of a free carbon, and we don't want that carbon to be free and hanging out. We want to be able to recycle it immediately. So we just pop it onto an oxygen and turn it into CO2, which is very easy to recycle. So during the intermediate stage, we break a bond, release electrons, so we have NADH, and then we also have a carbon, which we're going to recycle by combining it with an oxygen. So you'll also notice that another thing happens here. So we break this bond, and we convert a 3-carbon into a 2-carbon molecule. That 2-carbon molecule is called acetyl. But then we also add or pop on a coenzyme. So we mentioned coenzymes a while ago when we were talking about enzymes. Let's see if I can find that slide. We talked about impacts on enzyme function, and we talked about coenzymes. So coenzymes are organic molecules that can help a chemical reaction take place. So why the heck do we tack on a coenzyme here? And the answer to that is because the next step after the intermediate stage is the Krebs cycle. I really hope that this reference is not outdated and that we still have some like Game of Thrones fans in here. Game of Thrones fans said like, remember season 1, but not to ruin anything. Everybody dies in that show, so I feel like I'm not ruining anything by, you know, showing somebody that's dead. In Game of Thrones, not in real life. But basically what happens during the intermediate stage is you take that 3-carbon pyruvate, you break this bond, release electrons, which is why we get some NADH. You release a carbon binding up with oxygen, which is why you get carbon dioxide. But also to the resultant 2-carbon organic compound, acetyl, you tack on a coenzyme. This coenzyme A, so our resultant organic compound is acetyl coenzyme A, or acetyl-CoA. So the Krebs cycle is a pretty crazy thing, as we will see a picture of it here in a minute, but I don't want you to get overwhelmed because you don't have to memorize it. But you do tack on that coenzyme, and the reason you do that is because the Krebs cycle is a lot, right? And so you want something to kind of help that reaction proceed, and coenzymes help reactions proceed. So you're basically, by tacking on a coenzyme to the product of the intermediate stage, what you're doing is sort of just like making sure that the Krebs cycle will take place, right? So looking at the intermediate stage, so up here we have our energy tally. During glycolysis, we got 2 ATP via substrate level phosphorylation. We also got 2 NADH, which will be eventually used for oxidative phosphorylation. And then during the intermediate cycle, we got some NADH. We got 2 NADH. Why did we get 2? Right? So this is showing what's happening to 1 pyruvate, but remember, there's 2 pyruvates. And then this reaction happens to both of them. So technically after the intermediate stage, right, where we could just have, let's see if I can do it. Group. Technically after the intermediate stage, we could say that it looks, you know, like this, because we have 2 pyruvates entering. The same thing happens to them both, so we get double the products, which is why you see 2 pyruvates enter, utilize 2 oxygen, 2 coenzymes, and our products are 2 coenzyme A, or acetyl-CoAs, 2 carbon dioxides, 1 per carbon that popped off, and then 2 NADH, 1 per pyruvate being oxidized. All right, are we ready? We feel good. So we're leaving the intermediate stage. What we're tracking here is that acetyl-CoA. So brace yourself. The next slide is crazy. As you can imagine, it's the Krebs cycle. So I cannot make it not crazy. But the biggest thing here is don't get overwhelmed, because I'm going to tell you exactly what you need to know from it. So don't worry. Okay, not that crazy. Okay, so this is showing the Krebs cycle. I do want to reiterate the same thing that on here is you're looking at the Krebs cycle happening to 1 acetyl-CoA. But remember, because we broke glucose into 2 pyruvates, and then we broke that into 2 acetyl-CoA, this is happening to both of these. So this is technically happening twice. Okay. I guess it's happening once, but there's 2 reactants going in. Okay, so the Krebs cycle is sometimes also called the TCA cycle, and sometimes it's called the citric acid cycle. That all refers to a very important molecule, and that is a recyclable molecule called oxaloacetate. And the reason that's so important is because you can see, I drew this in just for continuity, but it's actually repetitive. You can see acetyl-CoA at the top of this cycle. So what enters the cycle is our product from the intermediate stage, which is acetyl-CoA. And what happens is acetyl-CoA, actually, which is a 2-carbon compound, actually combines with a 4-carbon compound, which kind of maintains in the cycle, called oxaloacetate. When it combines acetyl-CoA plus oxaloacetate, it forms another organic molecule called citrate. And citrate is a 6-carbon compound. So what we did here, why do we do this? You can see, obviously, like when we have just acetyl-CoA, that's just like we have one bond. So if we were to break that bond, like it would only give us a little bit of electrons. So the benefit of this, I'm so sorry, I haven't had coffee yet. So the benefit of this is that if we combine this 2-carbon molecule with a 4-carbon molecule and generate a 6-carbon molecule, all of a sudden we have way more bonds to break. If we have more bonds to break, we have more electrons to release, and we have the capability of making more ATP. And you can see that's just what we do. So what happens is we break these bonds, release electrons. You can see that we get some NADH. We get some more NADH. You'll notice we have 2 carbons released, which means we generate 2 carbon dioxides. 2 carbons went in, 2 carbons come out as carbon dioxide. So we generate some NAD, release electrons, generate some NAD. Hey, what's this? We get a little ATP via substrate level phosphorylation. Okay, and then we keep going. Notice we went from 6 to 5 to 4. For the first time, we're releasing electrons, and instead of using NADH, we're using FADH too. Same thing, they're carrying electrons. Man, we get some more electrons released. So here's the deal with the Krebs cycle. You do not need to memorize all of these things. Citrate, isocitrate, ketoglutarate, succinate, fumarate, malate. You may recognize some of those, right, as vitamins. But now you see why those are important. But all I want you to know is the reactants and the products. Yeah, that's all I want you to know. I want you to know the energy tally. So you can see that what goes into the Krebs cycle is this acetyl-CoA. And what's coming out, because 2 carbons are coming in, but we have 2 of those molecules, so technically 4 carbons are coming in. You have 4 carbons released. That binds with oxygen. Oxygen is required for the Krebs cycle. And then also per acetyl-CoA, you get 1, 2, 3 NADH, because you have 2 acetyl-CoAs, that gives you a total of 6 NADH. You get 1 FADH per acetyl-CoA, because you have 2 acetyl-CoAs, that means a total of 2 FADH. And you also get 1 ATP per acetyl-CoA, but because you have 2 acetyl-CoAs, that gives you 2 ATP. So that's all I want you to know for the Krebs cycle. You don't need to know all those steps, that's for a different class. But what I do want you to know is you get a ton, 6 NADH, 2 FADH, and 2 ATP. So we have so far completely, after the Krebs cycle, we have completely oxidized our organic molecule. We don't have any more of the organic molecule. And we have one more step of cellular respiration, and that's the electron transport chain. So you look at our energy tally, we have a little bit of ATP. So far we've generated 4, but we have a ton of electrons. 2 NADH, 2 NADH, 6 NADH, 2 FADH. And we're gonna send them to an electron transport chain. So what I wanna show you, I'm jumping ahead a slide if you're following along, but what I wanna show you is how those NADH and FADH make ATP. So NADH goes to the electron transport chain, and like we mentioned, it is oxidized. So it releases electrons. It also releases its hydrogens. That's where we get the regeneration of NAD+. That's again why with fermentation, why we have to have pyruvate accept electrons, because we have to regenerate NAD+. With cellular respiration, that regeneration of NAD+ occurs at the electron transport chain. But what happens is all of those electron carriers we've generated through cellular respiration, they come to the electron transport chain and they release their electrons and hydrogens. there's a big difference between NADH and FADH when they get to the electron transport chain. So the electron transport chain, you can kind of see it in this picture. So this is just looking at a membrane. The electron transport chain is made of 3 hydrogen pumps that are fueled by electrons. And at the end of that, you have ATP synthase. NADH, when it comes to the electron transport chain, is able to use an electron to pump 3 hydrogens across that membrane. So a very important thing to note is kind of what we talked a little bit about a while ago when we introduced oxidative phosphorylation. And that's that because every 1 hydrogen flying through ATP synthase will fuel the production of ATP, if NADH is able to fuel 1, 2, 3 hydrogens across this membrane, that means that NADH technically will be able to contribute to 1, 2, 3 ATP being generated. So that's why we say at the top, every 1 NADH will lead to the production of 3 ATP. FADH also is pretty good, but not as good as NADH. FADH, with every electron it releases, will be able to pump 2 hydrogens across that membrane. But again, because we have that ratio, 1 hydrogen equals 1 ATP, if FADH can pump 2 hydrogens across that membrane, that means that FADH coming to the electron transport chain will give us 1, 2 ATP. So I'm going to scroll ahead for a minute because I want to show you this. This is a very important chart for cellular respiration. So cellular respiration, this is kind of looking at the energy totals for all of those stages. You'll notice the electron transport chain is not listed here because what we're looking here is energy totals resulting from these different kind of major catabolic steps. So we have glycolysis, which does require 2 ATP, but it does generate 4 ATP via substrate level phosphorylation. But we say just a net of 2 because it required 2. Also during glycolysis, we got 2 NADH. And you'll see here that I have 2 NADH converted to 6 ATP. Again, that is because every 1 NADH will generate 3 ATP. So if we have 2 NADH, we say 2 times 3, that will give us 6 ATP. The intermediate stage where we broke pyruvate into acetyl-CoA enzyme A, that also gave us 2 NADH, so 2 times 3 is 6 ATP. And then we get to the Krebs cycle, which is insane. The Krebs cycle gave us 2 ATP via substrate level phosphorylation. Great, not a lot. But it gave us 6 NADH because every one of those NADH will generate 3 ATP. 6 times 3, that's 18 ATP. It also gave us 2 FADH, and every 1 FADH gives us 2 ATP, so 2 times 2 is 4 ATP. That gives us our total is 38 ATP. We used 2 ATP to get it started, okay. But we generated 38 ATP. For reference, let's think about this. Cellular respiration is a huge process. We just went through it. And it only requires 2 ATP. Okay, a lot of your chemical reactions will require some ATP, but on average, they require like 1 to 2 ATP. Cellular respiration requires 2 ATP to get started, and you generate 38 ATP. That means for every cellular respiration reaction you do, you generate enough energy to have 16 more cellular respiration reactions take place. That is a huge amplification. Okay, so I did pre-record some lectures. So what I would like you to do, because we're out of time, is look at those pre-recorded lectures. I went through the different steps of cellular respiration. I went through each of these, summarized it as well. I really do want you to know these energy totals, like how we get these. Know that this comes from substrate level phosphorylation and this comes from oxidative phosphorylation, the difference between those. And those supplemental lectures will also go through the rest of this. So they will go through kind of how other macromolecules can enter cellular respiration, how this relates to breathing. A really good question earlier was, can other molecules other than oxygen serve as final electron acceptors? Absolutely they can. It goes into that. Talks a little bit about fermentation and why fermentation takes place. And compares the three, and then of course there is a whole separate lecture that goes through photosynthesis. I will send out an announcement here soon. Once I get this lecture uploaded, I'll of course send an announcement again. And I'm also during that time going to announce that on Monday, I'm going to have a review session. So I'm going to record that review session so that everybody can access it. But I'm hoping that over the weekend, you'll kind of get through the Unit 3 exam, which will cover this stuff, and you'll get through the Learn Smarts, and you'll get through some other stuff, and that will basically kind of serve as a really good way for you to figure out what some of your questions are going into the final. So watch those out of class lectures, do the Learn Smarts, get that Unit 3 exam completed. You have the majority of the information you need for the Unit 3 exam. A lot of it's genetics, and then a lot of it's like cellular respiration, which we covered today. Some of it is photosynthesis, so be sure to watch that as well. But again, I have that discussion board. Please, please, please write questions in the discussion board. Go through the Unit 3 exam, have questions. Go through your previous exams and previous Connect homeworks. Those should all be open for you to look at and have questions. And what we'll do is on that review session on Monday, actually maybe what we could do is just have it during our normal class time on Tuesday. Because then it would just be scheduled, I guess. And then we can talk about all that then. So yeah, that's kind of what I'm thinking. So does the Unit 3 exam use Lockdown Browser? It does not. Thank you. I wanted to avoid any issues with that this time. So the Unit 3 exam uses Connect. So there is no Lockdown Browser for that. And then I didn't see the assignments for the lab listed as results discussion and revised protocol for lab 5 enzymes activities. Let me look at that, Erin, the lab results. Oh, it's not listed. We didn't do that. That was right when we had to move online. Lab 5 enzymes was our next mini report. And obviously we never had to do that. So yeah, sad. That would have been really fun. But yeah, we didn't get to, we did, I think, the preliminary lab 5 enzymes, but we didn't get to do the results discussion or revised protocol or anything like that because we didn't get to do those labs. So yeah. And then will the final exam use the Lockdown Browser? It won't. I'm trying to avoid as many technical issues for you all as possible with that. But both Unit 3 exam and the final exam are going to be using Connect, so your McGraw-Hill stuff. So it's the same as like your homework and the review sessions. You're going to get 2 attempts at it. It will take your best attempt. The only difference is that the final exam will be timed. So there's no lockdown browser. You can use your materials and everything like that, but the final exam will be timed. So that's the only difference. But you're going to have like 2 minutes, I think, per question. So still a significant amount of time. But that's the only thing, is the final is timed. The Unit 3 exam is not timed, but neither require lockdown browsers. So I wanted you to be able to access your notes, kind of go into it with that. Yeah, I don't know if you can access the ebook directly through Connect, but you can have another browser pulled up. So, yeah. And if there's any other questions, you know, let me know.